Well, ladies and gentlemen, our session is about to begin. Please have a nice seat. Thank you. Om Swastiastu. Assalamualaikum warahmatullahi wabarakatuh. Shalom sejahtera, shalom, namo buddhaya. Salam kebajikan, peace be upon you. Good morning, excellencies, distinguished guests, participants, ladies and gentlemen, which attend both in online and offline. I'm delighted to welcome you to the second day of the second International Seminar on Fish and Fishery Sciences 2023. Please give big round of applause to our togetherness here in the second International Seminar on the second day. Well, distinguished guests, participants, ladies and gentlemen, I'm kindly to inform you that today's agenda will be divided into 4 sessions, which are keynote speech, panel session, parallel session, and closing session. In closing ceremony, we will announce Best Award of the second International Seminar on Fish and Fishery Sciences 2023. Well, ladies and gentlemen, now we are entering the first session of today's seminar. That is to hear the keynote speech, which will be delivered by Dean of Faculty of Marine Science and Fisheries Udayana University. His last education is PhD at Chiba University, Japan. Well, ladies and gentlemen, please welcome Profesor Dr. Insinyur Iwayan Nuarse, MSI.
Good morning, distinguished participants, ladies and gentlemen, who are present in these rooms and joined online. My name is Iwayan Nuarsa from the Faculty of Marine Science and Fisheries, Udayana University. The title of my presentation is The Role of the Remote Sensing and GIS Technology in Fisheries. Firstly, I would like to talk about what is the remote sensing and GIS. Remote sensing is the acquisitions of information about the object or phenomena without making physical contact with the object, in contrast to in situ or on-site observation. And the Geographic Information System, or GIS, is a tool that we can use to analyze spatial data, not only from the remote sensing, but also from the various to produce the useful information. What is the contributions of the remote sensing and GIS in fisheries? By using the remote sensing, it doesn't mean we can see the fish directly from the space, or we can see the fish directly from the image. I don't think so. Remote sensing can detect and monitor the biophysical characteristic of the oceans or other aquatic resources and fish environments, including but not limited to. For the basic data, we can detect the sea surface temperature, sea surface height, sea surface salinity, sea surface chlorophyll, wind direction and speed, and others. And for the applications, remote sensing can be used for monitor and mappings of the seagrass, coral reefs, and others, mapping of bathymetry, mappings of spatial and temporal of upwellings, mapping of fishing grounds, and also mapping of the estimated potential fishing area in the future. This is the several examples of the remote sensing satellite imagery commonly used for fisheries and marine applications. For detect the sea surface temperature, we can use the NOAA, MODIS, and for the sea surface height, we can use the Sentinel, JSONs. For identify the sea surface salinity, we can use the SEX Aquarius. And for the winds, direction and speeds, we can use the Meteosat, Himawari. And for mappings, for the chlorophyll A, we can use Seaweeds, Aquamodis. And for the mapping of the seagrass and coral reef, we can use the Landsat. And for the mapping for the mangroves, we can use the Sentinel and Landsat as well. One of the important questions that need to be answered, that is whether the remote sensing is accurate compared with the field observations. A lot of research has been done to validate the accuracy of the remote sensing data by means of comparing the remote sensing data with the field observation. It is the example for the comparing the sea surface temperature obtaining from the remote sensing data with the field observation using the buoy data. The research show that the remote sensing data has the high accuracy compared to the field data. The coefficients of the determination show the accuracy and the root mean square show the error. In this picture, we can see the SST data validations with the Rama buoy and the SSH data with the tide data. In this picture, we can see the coefficient of the determinations is quite high, and then the root mean square error is quite low. This is the applications of the remote sensing data. This is the seasonal spatial distributions of Toba Lake surface water temperature and their trends based on Terra Modis from 2001 and 2021. In here, we can see the differences between the lake sea surface lake surface water temperature in the daytime, in the night time. That is that the lake surface temperature in the daytime is higher compared with the night time, and the lake surface water temperature in the month of the March, April, and May is the highest compared with the other session. In the image on the right, that is the trends of the lake surface water temperature during the 20 years. From this picture, we can see that there has been an increase in surface water temperature during 2000 years. I mean 20 years. This is the similar research in Poso Lakes. In the left side is the distributions of the lake surface water temperature in the daytimes and the night times, and in the right side is the differences between the daytimes and the night times. One of the most interesting of this research is that there has been a decrease in surface water temperature in Poso Lake during the 20 years. In contrast with the previous slide in Toba Lake. This is the interesting phenomena that needs to be the further research. Mangroves is one of the important habitats for the certain fish. Remote sensing can be used for mapping and monitoring of the density of the mangrove as well as the healthy of the mangrove. This is the applications of the sentinels to imagery for mapping mangrove density in Tahura Ngurah Rai. Red color indicates the high density and the green color is the low density. Remote sensing can also be used for mapping of bathymetry in shallow water. This is the study case mapping of the bathymetry in shallow water in the Benoa Bay, Indonesia. In this picture, we can see the darker blue color is the differ. This is the study about the integrations of the remote sensing and GIS to identify the suitability locations for fish apartments. In this study, use the three parameter, that is the bathymetry, the buffer from river outfall, and the seabed slopes. From this study, we can identify the best placements for the fish apartment placement. This is another application of the remote sensing relationship between the sea surface temperature with the tuna catch. The sea surface temperature obtaining from the remote sensing data and the tuna catch gets from the fisheries company. In this picture, we can see that higher sea surface temperature decrease tuna catch. This is the similar study relationship between the chlorophyll A with the tuna catch. In this study, we can say that the higher in chlorophyll A concentrations increase the tuna catch. Remote sensing data can also be used for mapping upwelling area distribution. This is the case study of analysis of upwellings area distribution pattern in South Indonesia using the MODES image. Using the three parameter, winds, sea surface temperature, and the chlorophyll A. From this study, we can say that the requirements for the strong upwellings are SST less than 27.66 degrees Celsius, chlorophyll A greater than 0.35 milligram per meter cubic, and speed and wind speed greater than 5.06 meter per second. Based on SST chlorophyll and the wind data, the strong upwellings occur on June, July, and August, and the peaks we found on August. This is the similar research was conducted in southern Java using the upwelling index. The results show that the upwellings occur in June until September, the peaks on August in mostly longitudes. The remote sensing can also be used for mappings of big size tuna fishing area. This study in the southern Java and Bali Island. This study used the two parameter, that is the sea surface temperature and the chlorophyll A. And this is the results of the fishing area. This is the fishing area maps of big tuna eyes. In here we can see that the blue dots indicate the locations of the fishing area, and the red and yellow circle is the positive and high tuna catch. And here we can see that the hikes and positive catch of the big size tuna mostly located in the fishing area. This is another application of the remote sensing to determine the habitat suitability index for big size tuna. In here, use the three parameter that is the sea surface temperature, sea surface chlorophyll, and sea surface hikes. In here, we can see the distributions and classifications of habitat suitability index. The red color indicates the high suitability, and the blue color indicates the low suitability. In previous study, in previous slides, the determinations of the fishing grounds is based on the past data. So the resulting of the fishing grounds is less useful for the fishermen. Therefore, we need to create the forecast fishing grounds maps for the future by forecasting the ocean parameter that determines the fishing ground. In this study, using the artificial intelligence, that is the deep learn approach, using the long short-term memory method. This is the result of the forecasting the distributions of the chlorophyll A in 2019 until 2020. In here we can see that the high concentrations of the chlorophyll A is found on September, October, and November, and June, July, and August, especially in the coastal area. This is the comparisons between the predicted and actual chlorophyll A. In here we can see the there are the synchronized about the predicted and actual chlorophyll A. Where is the correlations of the OR square is the heights. The coefficient of the determinations is in associations rings from the 0.83 until 0.96. In previous slides is the forecasting for the chlorophyll A, and here the forecasting of the SST. The picture in the left side is the actual SST, and the right side is the forecasting of the SST. And from the forecasting of the chlorophyll A and the SST A, we can create the map of the spatial distribution of the forecasting fishing grounds. This is for the future. This is the results of the forecasting of the fishing grounds of the big size tuna in the southern water of Java, Bali. In here we can see that the red color is the high potential for the fishing grounds, and the blue color indicate the low potential. This is the closing remarks of my presentations. The first, remote sensing and GIS has an important role in fisheries, especially in terms of providing information about the biophysical environments of fish, such as the sea surface temperature, sea surface height, sea surface salinity, chlorophyll A, wind direction, and speed. Remote sensing data has the high accuracy compared to the field observation data. Integrations of remote sensing and GIS can provide information about the ENSO, IOD, upwelling, fishing area, and other phenomena. That's all of my presentations. Thank you very much for your kind attention.
Well, thank you very much, Professor Iwayan Nuarso, for the keynote speech. We would like to ask you to stay on the stage because our organizing committee would like to present token of appreciation. Kindly be invited, Chairman of the Indonesian Ichthyology Society, Professor Dr. Chris Mono, and also Chairman of Organizing Committee, Ibu Dr. Nyomandati Pertami, to present the tokens to our keynote speaker. Well, thank you. Please kindly take your position for taking the pictures. Thank you very much, Professor Nuarsa, Professor Krismonon, and Ibu Dr. Dati Pertami. You may now take your seat. Once again, ladies and gentlemen, please give big round of applause for our keynote speaker, Professor Dr. Insinyur Iwayan Nuarsa, MSI. Well, distinguished guest participant, ladies and gentlemen, now let us start the second session, that is a panel session. So now, without further ado, we will start the panel session, and allow me to now invite our moderator. She is a lecturer at Faculty of Marine Science and Fisheries, Udayana University. Her last education is PhD at University of the Ryukyu, Japan. So please invite to the stage our moderator, Ibu Dr. Widyastuti Karim, MSI.
Good morning, everyone. Assalamualaikum warahmatullahi wabarakatuh. Om Swastiastu. Shalom, Namo Buddhaya. Welcome to the invited speaker session. Once again, my name is Widya Stuti. I'm your moderator for this session. In this session, we have Professor Hyun Woo Kim. I'm sorry, I cannot see clearly the PPT. He is a professor from Pukyong National University, South Korea. And his specialty is oceanography, aquaculture, and marine biology. So, good morning, Professor Kim. Can you hear me? I'm sorry, I cannot clearly hear your voice. Okay. Hello, Professor Kim.
Yeah.
Okay.
I can hear you.
Okay. Great. So I like to ask you whether you want to present your presentation directly.
Yes.
I mean by Zoom, or
Yeah,
you
by
will share
Zoom.
your...
I made a video clip.
Okay.
Yeah.
So prior to your talk, I'd like to introduce your profile first.
So, can you see me my screen? Can you see me?
I'm sorry, Professor, we haven't... Okay, yes. Now we can... Okay.
Okay.
Now we can see your presentation.
Okay, let's start.
So, excuse me, sir. So actually you have 25 minutes to deliver your talk. And
Okay.
then we have 10 minutes for discussion session. So time is yours.
Thank you. Uh, first of all, I'd like to thank special thanks to the faculty and the other staff members who prepared this special extension meeting. And I hope this meeting will enhance our further collaboration. Uh, Good morning, everyone. Nice to meet you. And first of all, I'd like to give my special thanks to the faculty and the other staff members who prepared these special international meetings. And I hope this meeting will enhance our further collaboration between the Udayana University and Bugyeong National University. Since we share the common interest in the fishery science, so collaboration between two universities will be helpful for both faculty members and the student. And today, I'd like to introduce or present a very short presentation about my current research. So basically, I majored in the genomics. So once I come back from the United States, and then I uh, had a job in the Department of Marine Biology. And at that time of moment, many professors asked me to apply this kind of my current genomic techniques to the currently used fishery science. So maybe 15 years of contribution of application of genomic tools to the fishery science now maybe became a very strong maybe tool to understand the fisheries or marine ecosystem, and a strong tool for the not just for the scientists and also for the policymakers and fishermen. Next slide. So food web structure is very complicated. So from the primal producer like a phytoplankton all the way to up to the top predator. So we have to understand for the scientific management of fisheries science. So, however, to understand the food web is not an easy task. So, in Korea, around the Korean peninsula, we spend millions and millions of dollars to understand the food web structure around the Korean peninsula. Then, however, operating the ship, as you know, it's highly expensive. So even though we spend a lot of money, and what we've been doing so far is collecting the zooplankton net, collecting zooplankton using the net, or taking water and using the microscope to analyze the zooplankton, phytoplankton. This kind of optical observations may be time consuming, and also sometimes many those microscopic organism has a different morphological characteristic. So even though recently they have some automatic system to analyze those species, but each country has their own their own morphological characteristics. So It's not a easy task, and it's very time consuming and labor consuming task. So, however, due to the recent development of next generation sequencing, the sequencing cost is dramatically dropped. So these days, maybe you can easily analyze your whole genome, which is the 3 billion base pair, just for the maybe $500. So due to those low cost, the technique we've been used for genomic human genomic analysis now can be applied for the environmental or fishery science. So that means those labors and time consuming task and the people, human resource and spaces and a lot of reagents can be saved. It's like like a big data and industrial revolution. Is also occurring in the fisheries. So those large sample bottles can be transformed into the small, microbials and stored in the -70. It can be stored, maybe several hundred years as long as we have a suitable condition. So it's maybe very fascinating to see some analyzed maybe 100, 200, years old water sample. Maybe some later scientists may be very excited to see any climate change or anthropogenic effect stored in the refrigerator. And also another good thing is that about the metabolic coding analysis is to the numerical factors, which is like a big data. If we take a look at this is just a one-time observation, right? But like a movie, if we analyze sequencing 100 times with the various time of series, it can be like a movie. So we can maybe have a more precise, more accurate data can be obtained from the metabolic coding with a relatively low cost. It doesn't necessarily mean no cost, which is a relatively low cost. As you know, the next generation sequencing is still costly for the single lab. However, the pipeline for the metabolic coding analysis is like this, just collecting sample and then constructing library and NGS analysis and identify species based on the bioinformatic analysis. Here's some example of what we've done so far using the metabolic coding or NGS analysis. First thing is to analyze the phytoplankton, which is the main primary producer in marine ecosystem. So we basically designed the primer based on the 23 plastid ribosomal RNA. That means we can specifically amplify maybe photosynthetic organism from the mixture of just the water. So after designing this one, this means we can quantify photosynthetic organism. So based on the 16S, which is a normally well-known bacterial component. So based on the quantitative PCR using the 16S and the 23S, we can see the both heterobacteria and then photosynthetic phytoplankton. So based on the qPCR, the result was astonishing. So we can exactly detect some spring phytoplankton blooming, and also we can see also some here. Take a look at this. As the sunlight is strong, different from Indonesia, Korea has a 4 season. So during the summer, and as the sunlight is strong, most of phytoplankton move to the 20 meter, not just the surface water. We can also see the migration of the phytoplankton according to the different season. So it's very straightforward. And then it's very useful to transform the traditionally known, maybe spectrophotometric method into the molecular biological quantification method. So in different depths, we were able to see from the south to the north, we can see some different amount of phytoplankton and the microorganism, and also relative amount of phytoplankton based on by comparing bacteria and phytoplankton. So you can see some certain part has higher amount of uh, bacteria and phytoplankton, because in April in Korea it's a spring, so water is becoming warmer from the south. And also we analyzed the bacteria and phytoplankton community based on the metabacoding analysis. So we can see the phytoplankton community is highly different from the surface water and 50 meters in depth. This means some water body is different from the surface water all the way down to the 50 to 100 meter. And in the June, as the water temperature increase, and then you can see some phytoplankton structure is wider, and but based on due to the stratification, the surface water has a narrower, and most phytoplankton are located from the 10 to maybe 20 meters in depth. So this is a very good methodology to see some phytoplankton community. Here's we also analyzed that phytoplankton community. Then we were able to see the cyanobacteria is the main photosynthetic primary producer in June, which is the summer in Korea. So due to as a result of a heat map, we can see the clear changes according to the season. There's a March and April at the surface water, and May, June has a different pattern. So what we found is from the beginning of May, the surface water photosynthesis mainly based on the cyanobacteria, not a large phytoplankton. However, during the winter time and early springtime, their phytoplankton community is based on the chlorophyta. So by analyzing this phytoplankton and microorganism structure, we can see the current status of the marine ecosystem. We also have some insight of primary productions of a marine ecosystem. Let's move to the environmental bio DNA. So when we just first introduced those techniques in Korea about 10 years ago, many people have been suspicious about these techniques. And then nowadays, many people just proclaim that I'm the major in the environmental biology, blah, blah, blah. So I came to realize that things changed in these days. So what it is eDNA? Many Indonesian researchers also know these, techniques. So like, like a previous study, maybe fish is continuously shedding of their DNA in the water. So water actually represent their fishy assemblies. So without any uh, direct catching or direct observation, by just the collecting water, we can see the fishy assemblies at that sampling site. This is the most revolutionary techniques. So, for instance, for the river, which is a small ecosystem, for the survey, here's a lot of, they caught a lot of fish. And this is another cause for the maybe destroying the habitat. But eDNA, just taking water. Okay. So there's no harmful effect on the ecosystem, especially regardless of the river, the ocean is much easier. No impact on the marine ecosystem. So methodology is overall same to the phytoplankton analysis. However, the primer is different. So primer and then maybe bioinformatic analysis is slightly different. But due to the previously renowned faculty members in University of Tokyo in Japan, the Professor Miya developed a PC-specific universal primer, which targeted 12S RNA rather than CO1, which is a well-known primer. However, I tried many different primer sets, but I found this primer is most reliable and feasible to understand the fish assemblage compared to the other like eco primer, any other fish specific primer have some cross activity, so they not successfully represented fishy species. When it comes to sea, some species identification, the MyFish is one of the reliable tools, I believe. So based on the MyFish system, we are able to see the 98 species, one Korean Gulf area, and the other Gulf area, we are able to see the 66 species. This is a pretty impressive that just the water sample, we were able to see the almost 100, 50 species based on just the water sampling. And then this is useful to see any endogenous species. On the left side, this is those are some one Gulf specific species. The other one is exclusively found in the other Gulf area. So I believe some MyFish-based metabarcoding analysis is really reliable and feasible tools to identify the fish diversity of a certain marine ecosystem. And based on the reader number, we are able to see where is the best place to detect this is to detect those one species. So, for example, two similar species, we were able to study some main differences. Okay, especially this species is exclusively found in some other Gulf area. And like phytoplankton analysis, this is a fish assemblage in Korea also shows strong seasonal variations. This is what we expected. And this graph shows some correlationship among the different species. Here, this species is cold water-like species, whereas the other 5 species is a warm water-like species. So we can see as the climate change, which species decreased and which species increased, we can detect at certain types of marine ecosystem. And also, based on the primer what we designed, we analyze ecdioplankton. This is also very interesting because without any sorting out any fish larvae and egg, right after fish plankton, zooplankton net, and then we can just homogenize and then using some ecdioplankton specific primer, and then we can identify the whole larvae and fish egg based on the PCR amplification. So we can see a lot of fish egg just simply based on the metabarcoding analysis. So we compared our result with the previously known result, but this is strongly similar to the previous result. And finally, I'd like to introduce some metabarcoding analysis can be analyzed, can be applied for the food web study. So this is a Antarctic toothfish, which is one of most expensive fish species commercially, and this is exclusively caught in the Antarctic area. But recently, many activists they argue that maybe catching of this fish may affect on the marine ecosystem. So we tested it. So it's been maybe 7 or 8 years to study the stomach contents. So previously they detect some fish species, but they cannot identify the species. So they asked me to analyze some species level prey items. So we conducted a metabarcoding as shown on the slide all around the Antarctic area. So what we found, we were able to isolate the 99 haplotypes and also 46 genera, which is about the 37 species. And what we found is that those Antarctic toothfish presumably or preferentially eat the fish. So this is a fishivorous species. And then those 4 species, including Kinobestiscus dwiti, Macros camel, Macros withani, are among the most abundant prey items in that ecosystem. Then we found that the prey item strongly represented the regional fish assemblage. So since this is a generalist predator and Antarctic toothfish can represent their fish assemblage. So for instance, 88.3, regardless of year, they have Kinobestiscus dwiti, the most abundant prey item, followed by the Macros camel. However, 58.4, which is the withani, is the most top predator. However, 88.1 is the Cryodraco antioticus. So this means, if we keep analyzing this database, so maybe if we catch this fish, we can see any impact on the marine ecosystem of their habitat. So we also propose these differences of prey item can be influenced by their physical oceanographic characteristics, for instance, gyre. So those two highly closely related to two species prey item have a unique geographical differences. So we can see that, okay, some Antarctic two species prey item can be affected by the some oceanographic influence. And also, we also found some shell and slope, which means water depths may impact on the prey items. For instance, based on the metabarcoding analysis, we can see the some preferential differences of prey fish. That means uh, at a certain water depths, okay, the Antarctic tooth fish preferentially eat this fish. So that means we have to understand maybe certain water depths, and then if we catch the fish at certain water depths, that can significantly impact on the certain species. So maybe based on the statistical analysis, we can see that some maybe short here some water depths and the region are both among the important factors of Antarctic species. So we can provide the policymaker don't catch the uh, don't catch the fish inside of 10,000 1,000 meter of a slope region. So outside is less impact on the slope marine ecosystem. So basically, due to the time limit, I cannot introduce all of my research. However, this is all. If you have any question, I feel free to answer about this. Thank you.
Thank you, Professor Kim, for the fruitful talks. So now we move to the discussion session. So any question? Okay, first question. So actually, I'm waiting for another question. Okay. Okay. How about the committee? Should the should he come to the stage or? It's okay. I think maybe it's better to. Okay, Professor Kim, we have one question for you.
Good morning, Professor Kim. Hopefully you can hear me. Can you hear me, Professor Kim?
Yes, I
Yeah.
can hear you.
Yeah, thank you for the insightful presentations. A very short question. My name is Satya. You can call me Satya. A very short question from your presentations. As we all know, that's a very insightful presentation, especially for our participants, that mostly students, undergraduate students. My question is, as we know, EDN is a very useful, advanced methods for using for analyzing biodiversity. My question is, do we still need morphological approach for biodiversity assessment from your perspective?
Yeah, thank you for very good
Thank you.
questions. I think, can you hear me?
Yes, crystal clear.
Thank you for very good questions. I think both studies should be conducted. First thing to do is morphological analysis of zooplankton and ecdioplankton. Maybe eDNA cannot see any maybe developmental stage of ecdioplankton. Okay, so eDNA is more numerical. I think maybe the researchers and students should understand the eDNA can be presented as a numerical value. However, we still need some kind of morphological analysis also should be conducted. For example, like if we catch some exioplankton of tuna, but eDNA cannot describe how does the exioplankton developmental stage, things like that. Okay, so and also maybe different developmental stage of zooplankton, phytoplankton. So eDNA cannot answer all. Okay, so we should aware of that. No single technique can answer everything. We should collaborate each other. So we still need a strong collaboration between the morphological scientists and the eDNA scientists. Yes.
So in short, we still need to combine both of these methods for specific cases.
Sure.
Yeah,
We have a different purposes. Yes.
it's clear. Thank you, Prof. Kim.
Thank you.
Okay, questions? I think this is a very rare occasion, so you should ask something. Okay, Bapak, okay, please come to the stage. Professor Kim, we still have
Sure.
one question.
Okay, good morning, Mr. Kims.
Hi.
Hi, my name is Jusmono from Universitas Jambi, Department Biologi. My topic research about DNA barcoding, and the next this year I will research about ADNA. My
Good.
question: how many primer to detect water until research the manifest in your slides?
Oh, so you studying the fish eDNA?
Yes.
Okay. There are different primer for their own purposes. If you'd like to for just the species identification, so my fish, which is developed by the MIA, is the best. However, if you'd like to study more in-depth studies such as maybe population structure, you should use the different primer. So there's no single magic bullet to explain everything. So maybe it depends on the maybe your research purpose. Another one is we should know that the regional or local reference sequence database should be constructed before applying for the environmental DNA. Because until now, our species identification has been strongly dependent just on the morphological scientist. However, the same morphological uh, appearance doesn't guarantee that the same genetic information. For instance, maybe many maybe same species in the Surabaya and the Bali, Lombok has their own different genetic structure. So maybe Surabaya scientists make their own local or regional DNA sequence database. However, the Bali and Lombok and those researchers, it's better to construct their regional local DNA database. So this is more important because until now, the fisheries management have been based on the morphological analysis. Same species doesn't necessarily mean the same ecological, same fisheries meaning. Maybe, maybe even though same species, according to the genetic structure, they have a different, migration pattern, different spawning time. So maybe we should manage the any fisheries based on the DNA base, not a species base level. Can you understand?
Okay, I understand.
Yes.
Thank you, Mr. Kim.
Yep.
Thank you, Pak. So maybe to simplify, different primer for different purpose, if I'm not mistaken. So actually, we have one question from the chat box in the Zoom, Prof. Kim. So it's from Ibu Celia Hermawati. So if we have problem to identify until species level, I mean some of the OTU only detect until family level or unknown. Do you have any solution or suggestion for this case?
Yes. That's pretty good question. I think this question maybe come from those who had experience eDNA one time. First of all, like I mentioned before, please construct a local regional reference database first. And then due to the short fragment size of my fish is only 170 base pair. So sometimes like a rockfish and the fugu and some flatfish, so they have a low genetic variation level. So my fish cannot discriminate correctly just by those short sequence. So it's pretty strongly recommended to construct a regional local database, okay, to see any species level, okay. For instance, if you have 2 or 3 candidates with the same sequence, maybe for instance in your local uh, site. But however, as long as you have a local database, you know which is among the 3 potential species, you know which one is which. Is that an answer for you? And then
Uh,
also, I got another question from the chat. I think cross reactivity of the primer. Okay. So cross reactivity. Oh, I have a more. I think I have too many questions. I think it's another one is cross reactivity. So you have to set up some proper PCR condition. If you decrease the PCR temperature too low, you have a lot of cross reactivity. So the successful eDNA research should be based on the proper PCR condition. Okay, so my piece, even though they recommend the 50 uh, in Celsius, but I usually little bit increased my temperature because it's lower cross reactivity to the other species. And then even how the lots of fishes. Oh, eDNA. Sometimes some people said the eDNA produce higher number of species than directly catch. This is very reasonable because not a single catching method is perfect. Maybe auto troll and any other gillet, each catching method have their own preference. Okay, so eDNA, maybe if you find some paper which compare the fishing method and the eDNA catching rate, but uh, maybe eDNA is a much higher number of you know num fish species identified. But we should be very careful. Some people think that eDNA analysis is a simple task. This is not a reality because eDNA is a very tiny amount of trace, DNA from the seawater. So the environment, lab environment should be very restricted and then maybe highly controlled. so that there's no contamination, cross-contamination across the sample. So if there's no specifically designed facility, so you can easily see the cross-contamination of each water sample. Okay, so you can easily see some, marine fish from the lake sample, something like that. So maybe eDNA analysis should be conducted in very highly controlled um, lab condition. So, for instance, in my lab, uh, maybe sample filtration and DNA isolation and the PCR are all conducted in separate, different aerated room. And end of the experiment, we use the Clorox treatment so that they have no cross reactivity. So this should be very careful. So eDNA, one more time, I'd like to stress that the eDNA analysis is a very tightly, controlled experiment. Okay.
Okay, thank you, Professor Kim, for the interesting discussion and all the participants. I'm afraid we have very limited time, so we have to
Yes.
end this session. I mean, okay. So
Thank you.
thank you. To sum up, um, for the future research, so eDNA and e metabarcoding, environmental eDNA, is a powerful tools to identify our current environmental status for the both marine and freshwater and ecosystems. But of course, it's not a perfect tool. So just like what Professor Kim mentioned before, we need to more collaboration in this field. And also for the local, we are local researcher, we need to build or construct our local database. Okay, thank you again for your participation and your enthusiasm in this session. See you all at different session. Thank you very much.
Thank you.
Thank you.
Thank you very much, Professor Kim, for your excellent contribution, and also thank you, our moderator, Ibu Dr. Widya Stuti. We would like to ask you to stay on the stage, and also I would like to inform that Professor Kim, please kindly remain on your or your screen, because we would like to present token of appreciation. The first A certificate is given to Professor Hyun Woo Kim from Pukyong National University. Once again, thank you very much, Professor Kim from Pukyong National University. And also, next, kindly be invited Secretary General of the Indonesian Ichthyology Society, Bapak Dr. Charles Simanjuntak, and also Chairman of Organizing Committee, Ibu Dr. Nyomandati Pertami, to present the tokens to our moderator for the first panel session. Well, once again, please kindly take your position, and also I would like to invite Professor Kim to turn on your camera screen for taking the pictures. Well, thank you very much once again, Professor Kim from Pukyong National University. Thank you very much, our moderator. and also Bapak Dr. Charles and Ibu Dr. Dati Pertami. Well, Excellencies, distinguished guest participant, ladies and gentlemen, we are now still together on the second international seminar on fish and fishery sciences. And we are entering the next session. There is the second panel session where we will hear from two panelists who are experts in their respective fields. Our panel session will be moderated by a moderator. She is a project manager at Yayasan Biodiversitas Indonesia. Her last education is Master of Science in Master Program of Biology Postgraduate Program Udayana University. And she will guide the conversation and also invite questions from the audience or participants. So allow me now to invite our moderator. Please welcome Ibu Niputudian Pertiwi, MSI.
Thank you, Master of Ceremony. Good morning, all of the honorable speakers, distinguished participants and guests, ladies and gentlemen. First of all, I am Diputudian Pertiwi. I will be your moderator for today's sessions in the second international seminar on fish and fishery sciences, ISFFS 2023. In these sessions, we shall have two outstanding presentations from our prominent invited speakers, whom I believe shall enrich our insight and knowledge regarding the theme of this conference. Before we begin the presentations, allow me to introduce our first speaker, Dr. Samantha Cheng. Okay, Dr. Samantha Cheng, she is the Director of Conservation Evidence Global Science World Wildlife Fund, or WWF. She completed her PhD in University of California, Los Angeles, majoring in biology in the Department of Ecology and Evolutionary Biology. Dr. Samantha Cheng is also focusing her work on data visualization and database creation, monitoring and evaluations, evidence assessment. Good morning, Dr. Sam. Can you hear our voice here?
Yes, I can hear you. Can you hear me?
Yes, clearly. Okay. I would also like to introduce our second invited speaker, Professor Dr. Insinyur Lucky Adrianto, MSc. Professor Lucky is a policy analyst and researcher focusing on fisheries and aquatic resource governance. He is also a professor in Faculty of Fisheries and Marine Sciences, IPB University. Professor Lucky completed his doctoral degree at Kagoshima University, Japan, majoring in Fisheries and Marine Resource Policy. Is Professor Lucky already in the room? Good morning, Professor Lucky. It's nice to have you here.
Good morning, everyone. Thank you very much for inviting me.
Yes, thank you, Professor. And now, before the presentation begins, I would like to address that in these sessions, each speaker will have 25 minutes to present the topic and then continue with the panel discussion for 10 minutes. And without further ado, I would like to invite our first speaker, Dr. Samantha Cheng, who will be presenting the topic about From Squid to Impact, Evidence-Informed Conservation. Ladies and gentlemen, please welcome Dr. Samantha Cheng. Dr. Sam, the time is yours. Dr. Sam?
Thank you. Sorry, I had a mute challenge. Hello, everyone. Selamat pagi. It's nice to see you all virtually. Thank you for having me. I will just share my screen quickly. Okay. So I believe you should be able to see add that now. Thank you for the wonderful introduction, Diane. As Jan said, my name is Sam Cheng. I'm the Director for Conservation Evidence with the World Wildlife Fund's Global Science Team. And today I'm going to be talking about the journey from understanding squid biology and squid population to figure out how we create better systems in conservation and natural resource management to understand the impact of our work and to improve the effectiveness of management over time. So just to start, I want to talk a little bit about the World Wildlife Fund. So WWF has hundreds of offices around the world that form a network. And these offices work individually and together to really work to develop strategies, implement programs, and collaborate and partner with different countries and organizations and communities to get to a world where people and nature can thrive in harmony. And so the portfolio of WWF's work focus on 6 different areas: forests and freshwater, oceans and wildlife, and climate and food. And to do this work, the Global Science Team is a team of scientists that spans the entire network. And our aim is to deliver evidence-informed solutions so that we can conserve biodiversity, so we can halt environmental degradation, we can address climate change, and we ensure human well-being. So across these different areas, the Global Science Team is comprised of different scientific experts and support and technical expertise to really try and bring together the best available science, data, and data systems to address the pressing questions that we're trying to answer in all of these different fields. So my job at WWF, I seem to be stuck. There we go. So my job at WWF is across all of these different areas. My background is as a marine scientist, but the work I do really focuses on developing systems for monitoring, evaluation, and learning, and for improving the use of evidence in the way that we make decisions in conservation and natural resource management. So what is MEL then, or monitoring, evaluation, and learning? It first really focuses on trying to understand what data do we need to gather about how we're implementing different types of projects or programs, what happens after we implement those projects, and how it is leading to the change that we want to see for nature, climate, and people, whether we're working in fisheries management or in marine conservation, or in ecosystem restoration. The second part of monitoring, evaluation, and learning is the evaluation part of it. So how do we take that information that we've gathered through monitoring, through assessments, to really understand how effective are these projects towards achieving these objectives that we've set? And then perhaps the most important part about this process is making the time to actually learn from that information. So now that we know whether or not it's effective, how it actually might work in different places, how do we use that information to adapt our program, to adjust our strategies, and to define our priorities as the world changes, and to be able to adapt and be resilient to that? So organizations all have different types of MEL approaches and MEL practices. We try and draw from research. We try and work with local communities. We try and work with governments to make sure that the systems that we're setting up to understand the progress and the process and the impact of our work is sustainable. So in order to do that, we really do need systems for managing that knowledge. We need to really focus on and support collaboration and that dialogue. I really appreciated what Professor Kim said earlier, that one thing is not the answer to everything. We really need to focus on collaboration, and I'll build on some of those themes in this talk so that we can have learning that is inclusive, but also allows us to learn over time. So the second part of my job that I had mentioned is around evidence, both in the generation of evidence about the work that happens on the ground and how change is occurring in natural systems, and also in the use of evidence. How do we take all of that evidence that's being produced through research that exists in different types of knowledge, and bring it together in a way that is usable and relevant to decision makers. So evidence comes from lots of different places. It comes from research, it comes from project evaluations, it comes from working papers and reports that come out of organizations and governments and institutions. And it also comes from individuals and communities in the form of indigenous, traditional, and local knowledge. But we know that not all knowledge is of equal reliability or relevance to inform different types of decisions. So what we need to do is really bring that information together and to do it in inclusive and in transparent ways so that we can make sure that we're accounting for potential bias in that information, that we reduce the likelihood of failure in designing something that's based on maybe faulty or missing information, and that we really do in a way that allows us to learn about what is happening and learn from what has already been done. But I think it's one thing to think about what does the evidence say, and it's another thing about how does evidence actually factor into the decision-making process. We know that decisions are influenced by lots of different things, not just what the science or the data says. It depends on what we value as a society. What are the priorities perhaps for a government? It also relies on knowledge that maybe isn't published, that's more about experience, knowing what sort of issues and factors are, and it also depends on the context. So in order to get to the middle part, to evidence-informed practice, we need ways that really can bring together the best available information and knowledge and be able to really combine that with sharing and knowledge sharing and collaboration and dialogue, so we can actually make sense of that information in the context of a decision or a scenario. So at WWF, I lead the conservation evidence team. We sort of develop our efforts and our approach based on 3 different pillars. And I'll build on some of these throughout the talk and sort of explain how these manifest in practice. The first is really on ensuring that the evidence that we have is robust and it's credible, meaning it's grounded in that best available knowledge. It allows us to understand how change happens across different scales, but really is relevant to specific contexts. So for example, knowledge that about and research about fisheries management in Oregon, where I live in the US, may not be relevant for fisheries management, for example, in Bali and vice versa, but figuring out how those two might link together really comes into that second pillar on the bottom of collaboration and inclusion. I think there are lots of communities of practice out there in lots of fields in conservation and in natural resource management, lots of different types of practitioners, researchers, and actors here that have different perspectives. And so to bring those together requires making time for that dialogue and making time for that learning. But then practically, the tools and the approaches that we develop and the data sets we try and bring together, we need to make sure that they're practical and they're efficient. Oftentimes, we have really complex methods that are exciting, perhaps from a research perspective, or they're really exciting because they leverage new technology like artificial intelligence and machine learning. We have to make sure they're actually feasible for example, for one of our field teams who might be working in a place with low internet connectivity, and making sure that they really meet those teams where they're at with the right time and the right resources to be able to either use that information or collect that kind of information to understand their impact. So all of this coming together, how does this relate to what I said at the beginning, that this was talking about from squid to impact? So for me, I just wanted to talk a little bit about my own experience and journey from working as a researcher and pursuing my graduate degree, and sort of talking about that early career stage and how that's led to the work I do now with WWF. So when I was doing my graduate work, I was really focused on trying to understand how different types of genetic tools could help inform fisheries management. At the time, I really focused on population genetics and population genomics and trying to understand what kind of information that these kinds of methods could provide to inform planning and implementation of fisheries for long-term sustainability. So at what point could these tools let us know what to catch, when to catch it, where to catch it, and how much, right? Sort of focusing on those main areas of fisheries management understanding. And at the time when I was working primarily in the Indo-West Pacific, in the Coral Triangle, being based out of Indonesia, one thing we were seeing was that as finfish fisheries were crashing, there was more attention being paid to cephalopod fisheries and squid fisheries in particular. As you can see here on this graph, and starting in around 2000, that increasing capture of cephalopods. But one of the challenges for cephalopod fisheries management is that most species that were caught were only identified to that class level. So just to the cuttlefish, octopus, and squid. And squid make up the majority of cephalopod catch. But we also saw there was a large portion of catch that was coming in that was just unidentified. They were generic cephalopods. And so there are a lot of questions starting to emerge about what kind of species are we even catching? How can we use that information to understand fishing pressure? And where might we need to change the way we manage or modify the way we manage these fisheries to ensure their long-term sustainability? So I focused on lolliginid squids. These are nearshore neritic squids living over the continental shelf. And they typically have a single terminal reproductive event, after which the squid usually break down and die. So the fishing pressure tends to occur around these spawning aggregations. So understanding the characteristics of those spawning aggregations, what species were spawning, when they were spawning, where they were spawning, was really critical. And for the group that I worked on for Sepia-toothis, we knew there was a lot of potential cryptic species that were out there. Sepia-toothis is a widely distributed species. It occurs throughout the Pacific, Indian Oceans, as well as the Red Sea and the Mediterranean. And there hasn't been good consensus about how many species might be there or whether they are in fact all one. And within those species, what the population structure might be like, so we could better understand spatial and temporal management of those fisheries. So in order to do this work, I used a lot of different methods, both field sampling as well as sampling in markets, working with collaborators at the Universitas Siakuala as well as Universitas Udayana, to both take morphometric measurements, to conduct field surveys, to look at behavior, as well as collect tissue samples, to look at population genetic and genomic and phylogenetic analysis. Here, apologies for my awful looking face. And what we ended up finding was that for Sepia-toothis, they had very complex population patterns. Of the squid that we looked at in the areas we looked at in the Coral Triangle, there were two very similar species that occurred in the same place, that had sympatric distributions. They were found in the same habitats. But when we looked closer, we found that one of those species showed a population genetic pattern indicating that it was a very limited disperser. Whereas the other showed patterns of genetic connectivity patterns that were more closely similar to more mobile pelagic species. So really trying to get at these questions of what are the barriers to gene flow and to dispersal, and how can we use that information to better understand how we manage it. What this information told us was that these two species live in the same place. They likely are harvested in the same way. They are often probably targeted by the same fishery, and they're really hard to tell apart morphologically. But we know that they have probably very different life histories, which would mean they would have been managed in different ways. So that's a lot of really compelling information that we thought was really important to communicate to the various different agencies and groups that were working in cephalopod fisheries management across the Coral Triangle. But as we started to talk with different management agencies and representatives, we quickly realized that there's a lot more information that needs to be considered than just the genetic patterns. We needed to be able to combine that data with the ecological, the biological, and the social data which we started to do by collecting information about behavior, about morphology, about potential diet. But we also needed to make sure that we had that information from all areas of their range. And so really required increasing our collaboration with other researchers and management agencies who are trying to look at these species. We also need to make sure that we were really meeting decision makers where they were at. So a lot of the decisions around fisheries management are not just informed by the scientific research, they're also informed by politics. They're also informed by different social values that people might have. So for example, for the work that I did on squid fisheries in California in the US, it's a very, very valuable fishery. It's of very high economic importance for the state and for the country. And so any information that might change the way that the squid were managed ran into a lot of negotiation with representatives from the fishers, representatives from the different commercial areas. And so it's trying to work through that. And what we discovered in Indonesia were two things. One, there was a lot of information that we were not connecting with in terms of the local and traditional knowledge of fishers and the knowledge about when different types of squid that corresponded to our cryptic species that we found were coming in, different characteristics that they could describe but were not captured in any way. But when it came down to it, the information that we needed wasn't just the genetics, because we needed to make sure that that information had somewhere to go, that someone could actually use it. And for a lot of managers and management agencies, they didn't have the capacity to act on that because they had low capacity to even check what squid were coming in and being landed. And so that information oftentimes just kind of sits there. So that really led me to the next portion of my career, because I started asking, well, what kind of information is most relevant to inform different types of conservation decisions when we know that decision makers have lots of other things they need to consider and lots of other challenges to be able to actually use this information. So first, how do we know when conservation is working, right? When the things that we're implementing are actually working, and how do we ensure this information can be used? So I just want to zoom back out for a minute and sort of put us in the context of today. We're at a time when action to address these global challenges for nature, climate, and people are extraordinarily pressing. They're very immediate, and there are lots of global frameworks and initiatives and commitments for climate change, the IPCC, for nature, for the Global Biodiversity Framework, and for people, for the Sustainable Development Goals. So if we want to understand whether or not our work is contributing to making progress, whether the work happening in our country is making contributions to this, to these global goals, we really need to understand what data we have and how that data can help us understand what is happening and what we do with that information. So we're at a really exciting time in science and in data production, where the production of data and knowledge is exponential. We have automated systems like satellites and remote sensing to almost collect data in real time about the world. We also know that scientific knowledge is doubling almost every 9 years. But at the same time, the types of decisions that we're trying to make, as I just described earlier, are increasingly complex. We're trying to achieve lots of different things together. And so that requires us to be more thoughtful about these different ways of knowing, trying to understand which perspectives need to be in the room to make those decisions, to decide what kind of information is actually going to be useful and relevant to these situations. So in the last bit of this talk, I'm just going to talk a bit about some of the work we've been doing to bring together that information. So going back to the beginning of this talk and trying to understand what do we know already and how can we use that to inform conservation and natural resource management. And the first area is really trying to support the synthesis, the review of this knowledge. So In the environmental field, there's been a rise in the use of systematic evidence synthesis. There's been a lot of interest in this really broad range of methods that have lots of strengths and weaknesses depending on what you want to use it for. But in general, these are robust and systematic methods. They can be socially inclusive, meaning that lots of different stakeholders can and should be engaged in the process of deciding what is the question that we're asking, that we want to find evidence to help us understand. They use systematic and transparent and reproducible methods, meaning we can update these reviews and syntheses over time as new information emerges. And they really try and capture a comprehensive or representative body of evidence to really capture all the elements that are important to understanding the question. So a lot of my work over the past 10 years has been focusing on understanding what evidence exists on the links between nature and people. The topic areas have really varied, but a lot of this work has been carried out with the Science for Nature and People Partnership, which is a partnership of 3 conservation NGOs, as well as with Conservation International, and then working with different groups from the World Bank to and other bilateral and multilateral donors, as well as with conservation organizations and government agencies to understand where do we have data gaps, where do we have knowledge gaps about some of these links that might indicate areas that are more uncertain, where we need to generate more information. Those are really research gaps that we need to work and partner with the research community to produce. And so through a lot of this work, we've built different ways of engaging with organizations to be able to use the insight from this work to inform where they prioritize their funding, where they prioritize monitoring and evaluation, and where they prioritize research. So if you're interested in using this kind of method, systematic maps, systematic reviews, and other types of rapid evidence methods, I would encourage you to look at the standards that are published by the Collaboration for Environmental Evidence. This is a network of centers around the world that set the methodological standards for conducting evidence syntheses, as well as feature syntheses that have been conducted and methods and guides through the Environmental Evidence Journal. So the other part of this work is not just conducting the syntheses, but helping others do it. So really trying to improve the efficiency by which we do this work. One is through a tool called Colander. It's a open source web-based machine learning platform for conducting document synthesis or literature reviews that helps users go through it faster to find those most relevant things sooner. And then the second is really helping people use the results of this synthesis. We have a number of different projects that have explored using user-centered design to visualize data from these maps so that people can explore them, filter them, and find the information that's most relevant to their area. And then the third is building a community of practice around using these different types of methods and developing practical guidance for those in conservation, development, and environmental organizations. This is the Rapid Evidence Assessment Methods group that's led by the EPA, along with a number of different organizations, the U.S. Environment Protection Agency. And then the other part of this sort of ecosystem of evidence use is trying to connect evidence producers or evidence generators, like researchers, with evidence users, not through synthesis, but by really creating that collaboration process and connecting people through it. So I talked at the beginning about the importance of monitoring impact, and oftentimes this conversation starts with, what should we be measuring? How should we measure it, right? Focusing on the indicators of change. But what's more important, that we've been trying to develop approaches and tools to help people really ask better questions. Try and understand, if we measure all this information, what are you going to do with it? What is it going to help you do? What is it going to help you do? And why do you need to know it? And part of this process is ensuring that we know who is asking the question, who's going to use the information. Are those the people, the right people in the room? Do we need to have other perspectives? And then we can really get up to that point of figuring out how we do it practically, figuring out how to do this cost efficiently and freeing up time so they can actually free up the time to adapt and learn. So part of the work we're doing now at WF is really trying to leverage artificial intelligence or remote sensing to help us figure out what we should be monitoring and how we should do it so that we can be as efficient as possible. So I'm just going to skip over this for a second here. I've talked a lot about evidence-informed practice, but I think the one thing that I wanted to provide as a key takeaway, or at least my key takeaway from my experience, is that getting to evidence-informed practice is an iterative process. And what's really key is that collaboration and that learning, because we are all working in these ecosystems, and we need to figure out ways that the work we're doing can actually sustain them and that our work can be sustained. And I think this is really important in the context of this study I wanted to share. This was some work that was done by a large collaboration of coral reef scientists, researchers around the world, because we wanted to understand who's doing research in coral reef science. Is it the people who are actively working in these places? Or, who's being represented? And what we found by looking at about 15 years of research in coral reef science is that only 4% of authors in 2003 were from non-OECD countries. And so based on the trajectory and that trend we saw, by 2046, only 50% of people publishing in coral reef science were expected to be from non-OECD countries, meaning the field is still really dominated by research emerging just from a few places around the world. But that's an issue, because the coral reefs around the world oftentimes do not occur in those places. And so there's a mismatch between where the research is happening and the research funding potentially is coming from and where the research is actually conducted. And this is not, in my perspective, is not a sustainable way of conducting research. It's not a sustainable way of informing conservation and natural resource management, because there is a disconnect between where the decisions are being made and the communities that are affected and the researchers who are doing it. Not to say that there aren't connections to be made. Thus, my concluding point here is that it's really important, obviously, to use evidence in the way we make decisions, but part of that process is really connecting and convening those stakeholders and tools to make sure that the right collaborations are in place, the tools meet people's needs. So it's not just about what we do, but how we do it and who's involved. So I'm going to leave it at that. We'll take questions, I think, at the end, and I hopefully will be able to answer them. In the meantime, please put any questions in the chat in case I cannot stay for the panel. Thank you, everyone. Terima kasih.
Okay, thank you for the interesting and
Recording
very
stopped.
informative presentation, Dr. Sam. And we will have panel discussions at the end of the talk after our second speaker, and we really hope that you can be there at the panel discussion sessions. Can you hear my voice, Dr. Sam?
Yes, sorry.
Okay, Sam. Okay, now we will continue our session with our second invited speaker, Professor Dr. Insinyur Lucky Adrianto. As I mentioned before, Professor Lucky is a policy analyst and researcher focusing on fisheries and aquatic resource governance. He is also a professor in Faculty of Fisheries and Marine Sciences, IBB University, and he completed his doctoral degree at Kagoshima University, Japan. And his areas of expertise including social ecological system approach to aquatic resource, integrated coastal management, and also fisheries socio-economic. Good morning, Professor Lucky.
Good morning. Good morning. Thank you very much for joining with the very important seminar today. Thank you very much. Selamat pagi.
Okay, Professor Lucky. Now, ladies and gentlemen, we will please Professor Lucky, to give a presentation Professor Luki, the time is yours.
Okay, thank you, moderators. I would like to share, I think following up the last or some slides of Sam's previous presenters related to the connecting not only the genetic, but also perhaps the other information, including the human dimension of the physics management. So that following with the main topics of this year's seminar, that actually the keyword is the resilience of the fisheries and the blue economy. I'd like to share the perspective of the how to manage the resilience of the fisheries toward blue economy transformation, of course, and then specific in the case of Indonesian. And I would like to also using the socio-ecological system approach as now I'm very passionate to use this approach in managing the resilience of fisheries, especially in the context of Indonesia. I'll start with the what is the blue economy, what's going on with that. I think it's not that really many slides on that, but I just try to put some refreshment on that. And but then I will also follow in with the context of fisheries and blue economy transformation. And by using that first and second topics, I would like to continue with the managing resilience of fisheries, especially the small scale fisheries related to the blue economy in Indonesia. And I will end with some of closing remarks. But before I continue, I would like to also express my grateful thanks to the all of the organizers, especially the Indonesian Society of Ecology, University of Udayana, other collaborators to when I got the invitation to come and join online in this statistics seminar. I would like to express my grateful thanks. Thank you very much for that, the organizers. Well, I will start with this. As some mentioned, that actually the fisheries is not only the fish, yeah. Right? And that's why actually I very happy with the seminar or symposium on fish and fisheries, because without human dimension, it's not fisheries. It's only fish, then only fish. Using the fisheries or forestry, for example, or agriculture, actually is the human dimension. That's what actually human inclusive ecosystem is very important in the context of the managing the resilience of fisheries. And I think this is my starting point of view related to the sustainable of fisheries, especially in the context of tropical countries like Indonesia. So human is integral part of the fisheries. The second one, I would like to go to the blue economy. What's the blue economy? Well, in my perspective, there is ecosystem supply and ecosystem demand. So I think in the context of ecosystem science, there will be something like the aquatic ecosystem as the system, which is have like services, like for provisioning, cultural regulating and supporting. But then because of that service, there is a demand, and demand connected with the human dimension, or we call economic system. In that context, resilience is very important, because then when the demand is excess the supply, and then the problem is the sustainability. But then if that only supply without the demand, then also not efficient ecosystem. So that in that context, I think the balancing between the ecosystem supply and demand is very important in the context of the complex adaptive system like fisheries in our country. And some of the example in my research using that framework actually is the backbone of the blue economy in my perspective. For example, like blue economy itself is very debatable concept, but I think in this seminar, I would like to concern that blue economy is aquatic system-based economy which implement the sustainability principle. Because I don't want to go to the debatable things, but some groups mention that blue economy is the environmental paradigm, they extended the green economy, and some groups also mention that blue economy is the ocean-based economy. But and then I try to combine between those two paradigms. That blue economy is the aquatic system-based economy, which apply the sustainable principle. And this operational dividend that I use in the context of blue economy. And this is the sum of that I mentioned, some of the operational definition, ASEAN Declaration of Blue Economy. Next month, I would like to go to the Belitung for discussion with the ASEAN Blue Economy Group, for example, and they use something like this: that sustainable ocean, all economic related activities with the ocean, and something like that. And this is the blue economy spectrum, and fisheries is part of this, right? I mean, for example, like the first one, the first thing, and of the marine living resources is part of the blue economy. So blue economy is not only the fisheries, but without fisheries there is no blue economy. So that I think we are, as the fisheries scientists, actually would like to have like confidence that blue economy is on not only fisheries, but without fisheries there is no blue economy. And then based on that, I would like to start with the context of the fisheries and blue economy transformation in Indonesia. What's going on? So this is the context of the operational definition that I use, as I mentioned previously. A system of economy which based on aquatic ecosystem, not only ocean, but including also the inland and freshwater ecosystem economy. So in the context of that, I would like to say, for example, the aquatic as the capital. This is the main capital of blue economy. Without aquatic system, there is no blue economy. That's why actually from the climate, was that the pollution, ecosystem health is very important in the context of blue economy. But then the other aspect of the blue economy is the economical and socio-economic aspect. Ecosystem integrity, human inclusive, ecosystem governance, etc., including the innovation and as also some previously mentioned regarding to the innovation, technologies, and also the other knowledge of the ecosystem by the stakeholders. And in Indonesia right now, there is some transformation. Next July, I got information that Bappenas, our national agency for development plan, would launch the blue economy transformation for the next 2045. We call it Indonesian Emas, right? Indonesia Emas, the gold period of the Indonesian for the next 20 years, 2045, that the DNA of the 20 years futures actually is the blue economy. And this is actually the emerging versus established sectors in the context of blue economy. So as I mentioned in this, or I put in the context of this screen, fisheries also is a part of the very important sectors in the blue economy. And this is the context of Indonesian case. We know that there is also some countries, including our neighbors like Vietnam, Malaysia, as well as the Philippines, also in the context of ASEAN, for example, I involved in that ASEAN Blue Economic Platform. They also have like a platform on this blue economy, and fisheries always included in the context of this blue economy platform in the respective countries. And for the Indonesian transformation for the next 2045 transformation, this is the thing that again, fisheries is very important as the very important characteristic of the blue economy, but not only fisheries. We talk about the blue economy. So this is the thing that from the archipelagic vision of our country, and then come to the context of SDGs, not only 14, but maybe mainly 12, 13, 15, and other related SDGs. And then in the context of that, we need integrated approach related to the regulation, institution, and as well as budget. And then come to the strengthened maritime vision. And we already know from the government, for example, at least from Bappenas, that the new long-term development plan will be come to the context of the Nusantara vision. Nusantara is actually, in my perspective, so like archipelagic system thinking, archipelagic vision that Indonesia is more dominant of the aquatic ecosystem, but without land, there is no Nusantara. So land and aquatic, land and ocean interaction platform is very important in our country. That's why actually the vision of our country is actually blue economic vision, put in the context of Nusantara vision and mission for the next 2045. And back to the fisheries issues on blue economy. This is our partial context, right? What's that? 14 fisheries management area for marine, and then 11 for the marine, and then 14 for the inland fisheries. This one. So what's next after we have like this? This is very important to be considered in this seminar. That this is not only the empty space. We have resources, we have ecosystem, and human is part of that all of the space, marine as well as the inland fisheries management area. So our responsibility actually how to deal with the managing this sustainable of fisheries within this management area. So that's our responsibility. In that context, I think I put example on the context of marine fisheries management area. There are many hierarchy, from the ecological hierarchy to the context of the government hierarchy. There is also multi-stakeholder platform who is the most responsible actors related to the sustainable fisheries. Many of the stakeholders, as Sam also mentioned, the stakeholders involvement is very important. So this is the thing that we have to deal with the context of the governance. The fisheries governance is very important to manage the resilience of the fisheries in our country. For example, like this is my very recent quick analysis of the, for example, fisheries management area for marine, for example. I put some modified, what we call modified copy plot. Usually, actually, the standard stock assessment, meaning that the copy plot is only the BMSY and EMSY, for example. But in this context, I put not only the ratio of the utilization of the fish stock, but also the economic value as the y-axis. Then the x-axis actually is the ratio. If we talk about the 17 to the 2020, you know that actually we have like CAPMAN, yeah, Ministry Degree of the Stock Assessment 17, and then the last year we have the new stock assessment 2022. So when we combine, there is some changes of the what we call the resilience of the stock, because then in the red one more than 0.8 ratio, for example, because more than that, I think it's already dangerous. We know that actually the precautionary approach of the reference point is 0.8 as general what biological overfish as targeted also in the SDG 14. But then in the context of the economic value, I divide into the green and the yellow, meaning that the green is the more valuable and then the yellow is less valuable. If you can see, the dot of that is actually the 9 group of species in the context of the each FMAs. You can see all that, except 718, yeah, because the 718, we know that it's still problematic because after we received the new Permen 22, number 1922, there's no change in the stock. But then if the talk about the context of the economic value, then there is different. What I'm trying to say is that please look at it, the dot, that actually the all of the most of the group of species is the red and the yellow. Is very small dot, not less number of dot in the context of the green area, meaning that actually this is the resilience. Our physics resilience is in dangers, in my opinion. That's why actually we have to be careful with the context of this stock assessment or stock resilience in our perspective. So this is the thing that actually the government should consider this graph regarding to the quota, for example. There is no much rooms of the fish stock actually in the context of the economic as well as the ecological perspective. So this is the thing that we need managing the resource resilience in the context of our fisheries. And then how then put in the context of managing that resilience? And this is my last part of my talking as blue economy, fisheries transformation in blue economy, and then the resilience, and then how to manage the resilience. I introduced actually it's not on the new one, and maybe most of you already know this. What we call about the social ecological system approach. The framework, sorry in Bahasa, but actually the context of the social ecological system is that the view of that social system is part of the ecological system. So there is some social ecological connectivities and social ecological co-evolution in the context of the pillars. In that context, I would like to say that actually there are four as modified from Ostrom, for example, that is resource system, resource unit as ecological features. There is some ecological rule there, and then also the resource actors and resource governance as the social system, and there is some social rule there. That's why actually the genetic, as Sam mentioned, or Professor Kim, for example, mentioned on the context of the eDNA, connecting the context of resource system and resource unit. But then, not only that information we need in the fisheries management, we need social rules, we need social values, we need resource actors value, we need some governance system value in the context of interaction and outcome. In that context, I try to promote that with this social ecological system approach, the managing the resilience should involve two things, two systems, ecological system as well as the social system as the one integrated system, or in some literatures mentioned that intertwined system, complex adaptive system in the context of the fisheries in our country. And the SES approach already been applied in the context of aquatic system, in inland fisheries, and in marine fisheries, as you can see in the context of this diagram, very comprehensive, we call it heuristic model, actually in the context of this comprehensive model. People and that I'm very interested with the Sam just presented on SNAP, for example, like, yes, we have to connect between the nature and people. And for my perspective, I'm using SES tools or SES framework to connect between the nature and people, both in the context of the inland fishery system, for example, in the left side, as well as in the context of marine, in the context of the right side, for example, in this diagram, from the climate to the context of the resilience of the community in the coastal area. In that context, I will end my talks today in the context of the application of that in my lab. We have fisheries resources management lab in our faculty, and under the Department of Aquatic Resource Management as well. My colleague, Dr. Charles, is part of that. The young scientist in our department that's very engaged on this seminar. I will promote actually the connectivities, for example, like this multi-scale analysis of the lobster, for example. We try to develop this model as the first identification, for example, which one is more vulnerable. Based on that, then connect to the content of the resilience. Dealing with the vulnerabilities of the fisheries, then we have to manage in the context of the resilience, for example, in the future. So by identifying the connectivity of all of the system, then we can identify the gaps or the feedbacks between the social and ecological system to identify the sustainability strategies, or we call actually IMS, yeah, adaptive management strategies for the fishery. And the second, for example, case study I did with my PhD student, for example, in the context small-scale coral fisheries in Selayar Island in South Sulawesi, for example. We try to develop the systematic model of the coral ecological resilience overlay with the context of the livelihood resilience. So in that context, the livelihood-based governance is maximum 45%. And then the effective marine conservation management minimum is 40%, meaning that actually we cannot say that, for example, full 100% protection or zero protection is not like that. We have like more detailed science on, for example, putting the in the context of marine spatial planning, how much the percentage of the protected area related to the context of the ecological and socioeconomic resilience. We cannot just only depend on one ecological resilience without consider the economic or social resilience, for example, and how to combine with that. That the system thinking and system dynamic we put of that using that I mentioned the connectivity system diagram to put some ideas on how ecological and economic resilience for small scale fisheries in, for example, in this context is the coral fisheries in Selayar Island. So that's thing that I believe leadership is very important in doing the ocean governance, including the fisheries governance, and using this inclusive leadership, meaning that we have to govern or taking some ecological governance put together in the context of that social and economic governance as the one system, what we call the social ecological system governance, will be very important tools to lead our country as the leading archipelagic country, as not only the largest archipelagic country, but also the leading archipelagic countries for the next 2045 period of our next Indonesia Emas, but using the fisheries as the main indicators of the blue economy. Blue economy is not only fisheries, but without fisheries, there is no blue economy in our very largest archipelagic state, Indonesia, uh, our beloved country. Thank you very much, the moderator, for the times, and back to you, and hopefully that we can still have some fruitful discussion with Sam after this. Thank you very much.
Okay, thank you, Professor Luki, for the informative and comprehensive presentations. Very well, ladies and gentlemen. Now we have come to the panel discussion session. And because our conference is a hybrid seminar, for audience in this room, you can ask directly by raise your hand. And for the online participants, you can also ask directly by clicking the raise hand feature or write your questions in the chat box. Please mention your name and state to which presenter you address your questions. So is there any questions for our speaker? For the participants in this room? So because there's still no one asked in this room, so we move to the questions in the chat box. So there's one question from Ibnu Sahidir to Dr. Sam. So I will read the questions. Dear Dr. Sam, this may be a little bit out of context, but I would like to inquire about the World Wildlife Fund's WWF general perspective on shrimp farming worldwide at present. Additionally, I am curious if there are any specific views regarding Indonesia. Dr. Sam, please, you can answer the question.
Yeah, that's a really good question. And I wrote a little bit in the chat. You know, I think for WF, I can't speak to whether we have a global position about all types of shrimp farming. But, you know, our strategies really do focus on sustainable aquaculture. You know, as we just heard from, I think, the many talks and the theme of this conference, shrimp are important, both for the shrimp themselves, their role in the ecosystem, and for the people who depend on them for their livelihoods and for the broader economy. And so what a lot of the work that we do as an organization does focus on sustainable aquaculture and focuses on the drivers of unsustainable shrimp farming. So some of the topics that just came up, right, with the social ecological systems and addressing governance, addressing rights around lands, addressing sustainable livelihoods, are all aspects of addressing that sustainable shrimp farming question. So I think me personally, you know, I think the way that I see it, we need to make sure that the work we're doing in conservation and natural resource management really tries to both meet us at the reality where people are at and what's feasible, but also with the eye of trying things out now so that we can work towards something better in the future. So, you know, shrimp farming is a particularly damaging practice for the environment, but we need to figure out how we address it in a way that doesn't do equal damage on human livelihoods.
Okay, thank you for the answer, Dr. Sam. So we will continue with the other questions. Is there any questions here from the participant in the room? For two of our speakers? Okay, there's one question here. Please, you can state your name and to whom you address your questions.
Okay, my name is
Please stand up. Yeah.
my name is Diko Mulya Subekti from Equity Relationship Management 2020. Permission to ask, what are the strategies that can be used to implement the blue economy concept, and what is the strategy for optimizing the potential of the blue economy? Thank you.
Okay, so the question is for Professor Luki, right?
Right. Yeah.
Yeah, Professor Luki, is the questions clear to you? Okay, yeah, please, you may answer the question. The committee, yeah, the question.
Yeah,
The
sorry,
committee,
because the
sorry.
administration actually doesn't allow me to unmute. I think that procedure is very difficult. So I think sometimes difficult to unmute myself. So thank you very much for unmute the thing. Yes, that's very challenging question, I think, in the context of the implementation of the blue economy. As I mentioned previously, blue economy is very not new. Yeah, blue economy is very common already in our country, because our country already the aquatic, quote unquote, yeah, aquatic and ocean dominated countries. So for the long history at Sriwijaya or Majapahit or Demak or something like that in the ancient kingdom already doing the blue economy actually, because the trade, for example, the mercantile, the trade between the Sriwijaya go to the Majapahit in Java, and then from Java go to the contact of Bali, Moluccas, including the Philippines, our neighbor country. All of that's already blue economy. That's why actually the blue economy is not really new in our country. But then, as I mentioned, the blue economy is emerging economics platform. Emerging meaning that we already have all the blue economy, but then we need some refreshment. We need some new spirit that actually Indonesia is the largest agricultural state. In the context of Indonesia, I mean, I'm not talking that the other blue country doesn't have the similar spirit, but then the strategy that I would like to mention is not right now. Already in the Indonesian government try to develop. We will develop the roadmap of the blue economy that now already prepared by the government. But not only the context of the roadmap or development plan, we also consider the context of the social ecological system. What is the really important of blue economy? Aquatic ecosystem, right? What is the challenge and threat for the aquatic ecosystem? We have to defend on that, because we thought the very healthy, or I think healthy ecosystem, there is no blue economy. In that context, a strategy is not only the development plan in the context of economic point of view, but also the context of the ecosystem governance. So my straightforward answer for the very strategic and challenging question of the audience is actually, let's waiting for the roadmap, waiting for the government plans of the blue economy, and after that, let's do let's what that things about the implementation strategy of that. For example, the three things, yeah, regulation perspective, we call regulation framework. The second one is actually the institutional framework, and then the last one is actually the financial framework. In these three strategic implementation scheme is very important for our country. Regulation. What is the regulation that we need to put some more powerful blue economy? The second one, institutional framework. Many of the institutions actually coming to the context of the blue economy, and who's the dirigent, who is the leaders of the context of blue economy movement. This is also the second strategy that we need to be considered. Last one, the blue financing strategy, the budget framework. Who will be finance the blue economy? From where actually the finance sources. In that context, the three pillar of strategy, regulation strategy, institutional strategy, as well as the finance strategy will be very important for our country related to the context of blue economy. I think we can discuss later on with the three pillars of strategy for each strategy, for the more detailed strategy of that. We can discuss in the later at the next or the other time for that. Thank you very much for the next for the question.
Okay, thank you, Professor Luki. I think we still have time for one more question. So I will read one questions to Dr. Sam from Rahmawati. So the question is, I would like to ask about the conservation practice that has been done by WWF or related government to cope genetically changes in squid, which is caused by increasing capture activity. Dr. Sam, you may answer the questions. Thank you. Sorry, Dr. Sam.
I had to unmute. Sorry.
Okay.
Thank you. Yes, this is a really great question, and I think this gets back at this understanding the whole system, right? So genetic changes that are wrought by fishing pressure come from lots of different places, and so if we want to address the genetic changes, we need to address these bigger drivers of fishing pressure. So, there's that part of it. But I think in general, when we talk about the explicit actions of WWF or other conservation organizations or of government agencies, I do think a lot of fisheries management and the work that's happening there to support it does focus on adaptive capacity in general. That adaptive capacity isn't just defined by genetic diversity or rapid changes in genetic composition and what those changes might be from, but focuses more on trying to ensure the overall health of that particular population or of that particular species. So I think this gets back to sort of that movement in natural resource management around ecosystem-based management, around sort of whole systems management, to look at these things as holistic whole and take an integrative approach. I think obviously we could be using a lot more genetic data and trying to understand changes to connectivity patterns, changes to both the demographic and genetic connectivity, and we are starting to see that. I think particularly in some land-based work in wildlife corridors, but I have not seen it to the same degree in marine systems, and particularly fishery systems.
Okay, thank you for the answer of Dr. Sam. So apparently there still have some questions left, but unfortunately, due to the limited time, we need to end our discussion session for today. And so, ladies and gentlemen, we finally have come to the end of this session, and I would like to say thank you to our distinguished speaker, Dr. Samantha Cheng and Professor Luki Adrianto, for a very interesting and informative talk, and also to all participants for the active participation. Please give a round of applause for the invited speaker and for you all. Thank you and good morning.
Thank you very much once again to Dr. Samantha from WWF USA and also Prof. Luki from FPIK IPB Indonesia, and also thank you all moderator, Ibu Dian Pertiwi MSI, and we would like to ask you to stay on the stage, and also Dr. Samantha and Professor Luki, please kindly stay on your screen, because we would like, our organizing committee would like to present token of appreciation. The first e-certificate is given to Dr. Samantha from WWF USA. Once again, thank you very much, Dr. Samantha Cheng, from WWF USA. The second e-certificate is given to Professor Dr. Insinyur Luki Andrianto MSc from FPIK IPB Indonesia. Once again, thank you very much, Professor Lucky Andrianto. And kindly be invited Vice Chairman of the Indonesian Ethology Society, Professor Dr. Insinyur Jumanto MSc, and also Chairman of Organizing Committee Ibu Dr. Nyoman Dati Pertami to present the tokens to our moderator. Well, for the next, please kindly take your position for taking the picture, and also I would like to invite Dr. Samantha and Professor Lucky to take a position for taking the pictures. Well, once again, ladies and gentlemen, please give big round of applause to our panelists and also moderator. Thank you very much, Dr. Samantha and Professor Lucky. Thank you very much, Ibu Dian Pertiwi as our moderator, and also Professor Jumanto and Ibu Dr. Dati Pertami. Well, distinguished guest participant, ladies and gentlemen, the next session will be a parallel session 3, which will be provided on the second floor of this building. And I'm kindly invite online participant to be on our breakout room to join the parallel session 3. And after the parallel session 3, we would like to invite you to enjoy the lunch, which will be provided in front of this main room. And after lunch, we will start the fourth session, which is the poster presentation, also in front of this main room. And our last agenda today is closing ceremony, which will be provided in this main room. So after fourth session, which is the poster presentation, please kindly back to this main room to continue our last agenda. Thank you.
offline poster, please go to the poster location immediately because the offline poster session will start soon. Thank you.
Okay, thank you. Good afternoon. I would like to presenting as my research. The topic is my research mangrove biomass sequestration in Benua Bay. The background is my research is global warming has been believed to be one of source of disaster. Global warming is a cause of imbalance in ecosystem as a result of increased gas emission. Gas emission is the last decades have double over from 1,500 million ton per year to 2,900 million ton per year. The extensive mangrove in Bali in Benua Bay, with an area about 1,300 hectare or 30% of area mangrove in Bali. The ability of mangrove in carbon absorption and storage is 3 times greater than other forests. Yeah. And ability of mangrove in carbon absorption and storage is indicated in biomass value. This research conducted in Benua Bay by on to time, yeah, and the time, the first time in Augustus 2022, and second time in March 2023. And the data collect award species identification, tree density, sapling density, total diameter, and canopy cover. And result and this result and conclusion is my research is 6 species were found in the area of study, and then the biomassa, yeah, was found in the second monitoring of for 2021 100 640 ton per hectare, and then was found increase the average of biomassa for 6 months is 1,315 ton per year per hectare, and then the increase in biomass directly proportional with increase in diameter at debris height or dBH. Those my presentation. Okay, thank you.
Michael, you have 5 minutes maximum to present your work. Are you ready to present your work?
Good afternoon. Thank you for the opportunity to present. Actually, this is my student works with the title Morphological Character and Molecular Identification of Threadfin bream Nemipterus in Yogyakarta coastal waters. Threadfin bream, or we call curacy, is an important and potential fishery resource, and then high economy for fish commodity. And one of the challenges in the study about the diversity is Nemipterus, and there are many species of Nemipterus, but we cannot know what the exactly inhabit in a particular water. So we want to study about the Nemipterus, what kind of species in this waters in Yogyakarta coastal water. And the first sample we collected from September to December 2021 in the Baron fish landing ports. Then we conducted in the two methods. One is the morphological identification following the Russell and Ho in 2017, and each fish sample was measured 22 morphometric character. Then we also count 5 mestic characters, and therefore then we conducted a molecular identification following by DNA PCR method by direct sequencing with the target is cytochrome oxidase subunit 1. And the result and discussion by the morphological identification we found 2 species, namely Nemipterus fucillatus and Nemipterus gracilis, and the name is not common because we always call Nemipterus japonicus or Nemipterus virgatus, but we found only 2, Nemipterus fucillatus and Nemipterus gracilis, and we only found 2 species, 2 individuals that identified as Nemipterus gracilis, but mostly Nemipterus was Nemipterus fucillatus. So this is the picture figure for Nepterus succillatus and Nepterus garis, and the identification based on the morphology. And this is on the meristic character of Nemipterus coats and the Yogyakarta coastal water. You can see in this. This is our study. Then the compare with similar character by Russell and Ho. And this is for Nemnipterus gracilis. So I think there are only small differences between Nemnipterus succillatus and Nemnipterus gracilis. Maybe only on the number of squama in the linea lateralis. Then we conducted PCR, principal component analysis test analysis, and we found that the Nemnipterus succillatus and Nemnipterus gracilis shown the very similar based on the morphometric characters. Then after that we conducted a molecular identification, and this is the summary of morphological and molecular identification of Nemnipterus coats in Yogyakarta coastal waters. So based on the morphological identification, then the first sample identify as Nemnipterus gracilis, number 2 Nemnipterus succillatus, and number 3 Nemnipterus succillatus. But by molecular identification, actually the Nemnipterus gracilis was identified as Nemnipterus succillatus. So in this figure you can see in the molecular phylogenetic tree showing the genetic relationship among Nemnipterus and found in the Yogyakarta coastal water and other location, and also the outgroup Paracolopsis academy from Taiwan. As the summary or conclusion, we found that based on the molecular, only Nemnipterus succillatus found in the Yogyakarta coastal water. Thank you very much.
Check. Okay, thank you. My name is Fernanda. I'm from Fisheries. So I will tell you all about the ORP application in the shrimp farming. So water quality management is crucial in shrimp farming as it directly impacts the shrimp health and productivity. However, assessing water quality can be complex, requiring timely analysis and decision making for giving effective treatment. ORP as an indicator of a substance tendency to oxidize or reduce another substance shows promise as a parameter for summarizing water quality in shrimp farming. By having a single parameter that provides a summary of water quality status, we can facilitate efficient decision making and rapid action in response to the changes of the water quality itself. While ORP is commonly used for assessing the pond soil sediment, this study specifically focuses on the the ORP of water. So we collected the ORP data at 1 minute intervals over 60 days at our farm in Cipatujah, Tasikmalaya, by placing a stationary probe of ORP matter. And the results show that ORP values may not have a significant a direct correlation with the other parameters, and there's also found a complex scattered pattern for the before and after treatment analysis. In conclusion, ORP values are better suited for controlling and monitoring specific system rather than being used as a sole parameter for interpreting the overall pond dynamics. Thank you.
Okay, thank you so much. So this is actually a collaboration research between Universitas Brawijaya and also Ministry of Marine and Fisheries Affairs. So the title is ENSO and IOD Impact on Tuna Fisheries in South Indian Ocean. My name is Sambah. I'm from the Faculty of Fisheries and Marine Science, Universitas Brawijaya. So the urgencies of this study consists of three issues. The first one is currently Indonesia Planning Agency, or we call by Bappenas. Now they are preparing for the roadmap, Blue Economy roadmap for 2024, which is they have two targets. The first one is to increase economically the national income, to increase the national income from the maritime sector up to 17.5% from the fishery sector. And the second one, ecologically, we have to make sure that our fisheries resources is in sustainability term. And the second urgency is we know that we have a lack of data about the migration of pelagic fish, especially for the economically fish like tuna and also another species. Based on that urgencies, we know that Indonesia support more than 16% from the world total production from the tuna all over the world. So, and also based on the academic background, we know that the habitat of fish, or especially the habitat of pelagic fish, is indirectly influenced by not only oceanography factor, but also climate effect. We know that in the Pacific side, we have El Nino Southern Oscillation, and also in the southern part of equator, we have Indian Ocean Dipole. And indirectly, we believe that these both phenomena affect indirectly to our habitats, not only for tuna, but also maybe for the high migratory species like marine mammal. So our research aim is to know or to determine the effect of climate anomalies such as ENSO and IOD to the productivity of one of the species of tuna, yellowfin tuna, especially in Indian Ocean. So we collected more than 400 points of fishing ground. So around the area of Malang fishing port, or in the southern parts of East Java, we collected from 466 points of fishing ground, and we collected the catch data of yellowfin tuna from 2017 until 2021. And also we compare with the oceanographic parameter data such as sea surface temperature, chlorophyll A, and also we also applied NINO 3.4 data and also dipole mode index, and we analyze all the parameter using generalized additive model. And in the end of this research, we want to create a potential map or potential distribution map of the yellowfin tuna especially. So in conclude, from tuna catch dynamic, we will show the tuna catch dynamic for the years of 2017 until 2021, and also how about the dynamic or, variability of oceanographic parameters? Consists of SST, sea surface temperature, and also sea surface chlorophyll. And also we identify the ENSO and IOD during the period of 2017 and also 2021. And using the correlation and game analysis, we do some standard analysis to know which parameter that affect to the distribution of yellowfin tuna. And the final one, 2023, we want to produce a dynamic map of yellowfin tuna fishing ground. This is the result. So from the correlation analysis and also generalized additive model analysis, we found that the variability of SST and also SSC, sea surface chlorophyll, was influenced by IOD phenomena and not significantly influenced by the ENSO phenomena. And from the 15 GAM models, we found that the model consists of, or the combination of chlorophyll A data and also ENSO phenomena affect the dynamic of tuna catch. This is, we know this from the value of AIC and also DE value. This is some Hofmöller diagram that we know that the pattern of the SST and also the pattern of SSC is inverse. It means that sometimes the increasing of temperature followed by the decreasing of chlorophyll A and followed by the highest number of the cats. But the pattern is not so clear. So the result is not so significant, statistically. And also here we can see this one is the combination or the analysis between cats, yellowfin cats, and also the pool mode index. Almost similar what we found here, that almost the atmospheric parameter and also oceanographic parameter affect indirectly to the distribution of the tuna or to the distribution of the especially yellowfin tuna. In conclusion, the phenomena of climate anomalies and oceanographic condition in the water indirectly affect fish catch through the food chain process. It means there are a time lag between the increasing of oceanographic parameters and the productivity of the catch. Time lag, and based on our research 10 or 5 years ago, the time lags may be from 1 until 5 years. So we thank you so much to Ocean Color and also PSL NOAA for providing oceanographic data, and also NINO, that's 3.4, and also DMI data, and Pondok Dadap Fishing Port in Malang for supporting tuna catch data from 2017 until 2021, and also this research is a part of Hibah Penelitian Unggulan in our university. Thank you so much.
Yes, you can call me Rian. Rian. Yeah, Norita Vanda Oktarian. Yes, thank you. Yes. Okay. Okay. Okay, thank you. Thank you for the time and for the in this day I will to a presentation my mini review. This mini review is contribute for my dissertation, and the title is the potential of lactic acid bacteria or BAL to control histamine production in seafood product as a mini review. In effort to maintain the stability of demand of seafood consumption, the relevant authorities must be aware alarming increase in case of food poisoning. In this mini review, we are control for the histamine contamination in the fishery of product. As we know, histamine can be some disease can be like allergic if the human consumption, like fishery of tuna. And then, however, some of this way can trigger health problem. Some people can get allergen, like tongue can be allergenic, and then they need to eat antihistamine to again to from biological product which identically have a good efficiency and then easy to apply. So we need to find biological control like lactic acid bacteria. However, in the company, they have to they have a strategic to prevent the histamine, like a lower temperature, and then in the scientists we need to find one control like use lactic acid bacteria. Lactic acid bacteria, as we know, to produce histamine, some lactic acid bacteria also have histamine. You can see on the table, some of the lactic acid bacteria also produce histamine. So they can increase the histamine on the seafood product. So we need to select the lactic acid bacteria which no produce the histamine. So we can tandem with the bacteria BPH, bacteria produce histamine. So we can increase the we can to prevent the histamine, to control the histamine from the bacteria produce histamine. You can see on the table D, lactic bacteria acid have been highlighted in recent years for being a breakdown biogenic amine by producing amine oxide enzyme. So the species like Pediococcus and Plantarum have been isolated on my dissertation to suppress bacteria potential histamine. So if we can find more bacteria, lactic acid bacteria, we can use to control the bacteria histamine production in seafood contamination. Okay, in the last my presentation, the conclusion is histamine producing bacteria has been successfully isolated from various seafood products. In my dissertation, I have been finally isolation the bacteria potential histamine isolated from the yellowfin tuna and also from big eye. And then I try to put bacteria lactic acid on the histamine tuna, and then we are control the increase of histamine. I think that's all. Thank you for the time.
Yeah, thank you. All presenters have present their poster. Thank you again for all of the poster presenter. So today's session poster, we end this poster session for today. Thank you. Kasih range sih.
Well, good afternoon, ladies and gentlemen. Welcome back to the, thank you. Welcome back to the second international seminar on fish and fisheries sciences 2023. And now we are getting closer to the end of this seminar, but now we are entering to the door prize session. All right, it's time for the raffle draw where you can win some exciting prizes, and the draw is taken from the abstract number. Once again, the draw is taken from the abstract number. So ladies and gentlemen, please remember your own abstract number. We have two prizes in the first session of door prize to give away today, and also we have two prizes in the second session of door prize. And to raffle the draw, we will use spinner application. So the committee, please be ready with the spinner. All right, for the first winner, let us count down together in 3, 2, 1, go. All right, congratulations for the first winner. The number, the abstract number is FMG02. All right, we move on to the second prize. And let us count down together for the second prize in the first session of door prize. In 3, 2, 1, go. All right, congratulations for the second winner. The abstract number is FMG05. All right, ladies and gentlemen, the winner of this door prize will ask by the committee to claim the prize. So once again, please remember your abstract number. Well, ladies and gentlemen, before we move on to the next agenda for today, let us now enjoy a special performance. Once again, a special performance named Joget Bumbung. All right, be ready, ladies and gentlemen. Joget Bumbung is a traditional Balinese dance performance intended as entertainment for the community and also for tourists. The dance is one of the 9 Balinese dances proposed to be the World Cultural Heritage by UNESCO. The word joget is defined as movement or dance. Meanwhile, bumbung is a local term which means bamboo. This is a dance or movement accompanied by bamboo music. So ladies and gentlemen, please enjoy Joget Bumbung Dance. All right. Once again, ladies and gentlemen, please give big round of applause for the dancer. That was Joget Bumbung. Once again, Joget Bumbung is a traditional Balinese dance. And now we move on to the second session of door prize. I would like to ask the committee to raffle the draw. On the second session of door prize, we also have two prizes to for two winners. Okay, let me start to raffle the draw. In 3, 2, 1, go. All right, the first winner of the second session door prize is AGO58. And the last prize for the second session door prize, let we count down together in 3, 2, 1, go. All right, congratulations for FMG 15 or 15. Congratulations once again to the winner of our door prize. And now, ladies and gentlemen, as we informed you before, there will be the best award ceremony before the closing session. And now we come to announce the winners of each category. The first category is the best oral presenters. The best oral presenter in room 1 is Hilda Mardiana. Congratulations, Hilda Mardiana. The best oral presenter in room 2 goes to Nyoman Giri Putra. The next one, the best oral presenter in room 3 is Sekar Larasati. All right, the best oral presenter in room 4 is Saiful Ahmad Tauladani. Congratulations. And the last one, the best oral presenter in room 5 is Novalia Rahmawati. Congratulations once again to the best oral presenter, and I would like to invite the winner to come to the stage. Online. All right. Well, ladies and gentlemen, we move on to the next category. The next category is the best online poster presenter. The best poster presenter. The best poster presenter in room 1 is Diana Arfiati. The best poster presenter in room 2 is Woro Hastuti. Next, best poster presenter in room 3 is Henderite El Ohi. The best poster presenter in room 4 is Husna Husna. And the last one, best poster presenter in room 5 is Han Na Jang. Congratulations to all winners best poster presenter. The next one is the best offline poster presenter. The best offline poster presenter. This category goes to Abu Bakar Sambah. And the last category, ladies and gentlemen, this is the special award. Special award, Best Undergraduate Student Presenter. The best undergraduate student presenter goes to Nimade Dwiyanita Soka. Once again, Nimade Dwiyanita Soka. Once again, congratulations to all winner in Best Oral Presenter, Best Poster Presenter, Best Poster Presenter Offline, and also Best Student Presenter. Is Abu Bakar Sambah here? Okay, Abu Bakar Sambah, the Best Poster Presenter, please come to the stage. And also Nimade Dwiyanita Soka. And to present the appreciation, allow me to invite the Chairman of Organizing Committee, Ibu Dr. Nyomandati Pertami, once again to present a token of appreciation. Thank you and congratulations once again to Abu Bakar Sambah, Best Poster Presenter, and thank you also Ibu Dr. Dati Pertami. Well, ladies and gentlemen, we have now arrived at the last session of the second International Seminar on Fish and Fishery Sciences 2023. And to begin the closing ceremony, let us now hear a speech from the Dean of Marine Science and Fisheries, Udayana University. I would like to invite Professor Dr. Insinyur Iwayan Nuarse, MSI. The stage is yours.
Good afternoon, Excellencies, distinguished participants, ladies and gentlemen, who attending offline and online. In conclusions, the international seminar on peace and fisheries sciences has been an enriching and enlightening event, bringing together experts, researchers, and enthusiasts from around the world to delve into the fascinating realm on fish and fisheries. Throughout the seminars, we witnessed the exchange of valuable knowledge, cutting-edge research findings, and innovative approach in the field. The seminar has provided a platform for exploring diverse topics, including fish biology, aquaculture, fisheries management, conservation, and sustainable practices. The discussions and presentations have shed light on the challenge facing our aquatic ecosystem and the importance of responsible fisheries management for the future. By collaborating and sharing our experience, we have gained fresh insights into the complexities of the fish and fisheries sciences, discovering new methodologies, technologies, and strategies for sustainable fishing practice, habitat preservation, and the protections biodiversity. Furthermore, these seminars has fostered international cooperations, promoting global partnerships in research, conservation efforts, and knowledge disseminations. The connections made here will extend beyond the seminar, serving as foundations for continued collaborations and progress in the fish and fisheries science field. As we depart from the seminars, let us carry out the knowledge gained and the connections made to our respective fields and communities. Let us try to implement sustainable practices, advocate for responsible fisheries management, and contribute to the preservation and well-being of our aquatic ecosystem. I would like to express my gratitude to the organizers, speakers, presenters, sponsorship, and attendees for their valuable contributions, passionate discussions, and commitment to advancing fish and fisheries sciences. Together, we can make a positive difference in the world, ensuring the future abundance and sustainability of our aquatic resources. Thank you and best wishes for continued success in the next event. Thank you.
Well, thank you very much, Professor Nuarse, for the speech. And ladies and gentlemen, last but not least, I would like to invite the Vice Chairman of the Indonesian Ichthyology Society, Professor Dr. Insinyur Jumanto MSc, to deliver his closing remarks for all of us. Professor Jumanto, the stage is yours.
Bismillahirrahmanirrahim. Distinguished guests, esteemed participant, valued contributor, excellencies, ladies and gentlemen. Good afternoon. Assalamualaikum warahmatullahi wabarakatuh. Shalom, Om Swastiastu, Namo Buddhaya, Salam Kebajikan. Today, we gather here for closing ceremony of the International Seminar on Fish and Fisheries Sciences. It has been an extraordinary journey of knowledge of acting, collaboration, and exploration within fish and fisheries science. On the first day, 320 people attended the second international seminar on fish and fisheries science. Consists of 144 participants, 131 oral presentation, and 45 committees. On the last day, there were 161 participants, 74 oral presenters, 14 poster presentations, and 38 committees. Over the past 2 days, we have witnessed the remarkable dedication and passion you have brought to this seminar. Throughout this event, we have delved into a wide range of topics, from sustainable fisheries management to innovative aquaculture practices, from the impact on the climate change on fish population to the preservation of marine ecosystem. Our discussion have been enriching, enlightening, and thought-provoking, shedding light on the challenges and opportunities to the lie ahead. We extend our heartfelt appreciation to all the researchers, scientists, scholars, industry professionals, and policymakers who have shared their expertise and insight with us. Your invaluable contribution have significantly enriched our understanding on fish and fisheries sciences. The depth of the knowledge and the breadth of perspective shared during this seminar have been truly remarkable. We also express our deepest gratitude to the organizing committee, volunteers, and sponsors for their unwavering support and dedication. The tireless effort behind the science have ensured the smooth execution of this seminar, creating a conducive environment for meaningful exchange and collaborations. As we reflect upon the accomplishment of this seminar, it is essential to acknowledge the significance of international cooperation in addressing the pressing issues facing our ocean and aquatic resources. The challenges we face are global in nature, and they demand collective action and collaborative solution. Let us strive to build upon the connection made here, fostering partnership and networking, and will transcend borders and facilitate the exchange of knowledge and best practices. In closing, we want to emphasize that this seminar is not merely an endpoint, but a stepping stone toward further advancements in fish and fisheries sciences. The knowledge shared to ideas, spark, and the relationship forged during this event will undoubtedly pave the way for groundbreaking research, sustainable practices, and effective policies that will shape the future of our ocean. On behalf of the organizing committee, we extend our sincere appreciation to each and every one of you for your active participation and engagement in this seminar. We hope that the insights gained and the connection established will continue to inspire and propel us forward on our shared journey toward a better understanding and stewardship and of our fish and fisheries resources. Thank you all for your outstanding contribution, and we look forward for future collaboration and the relation of more sustainable and thriving future for fish and fishery sciences. See you in the next third ISFFS on 5 to 6 June 2025 in Yogyakarta City. And this city is very interesting for you to be there. Thank you. Assalamualaikum warahmatullahi wabarakatuh.
Well, thank you very much, Professor Jumanto, for your closing remarks. And this also marks the end of the second International Seminar on Fish and Fishery Sciences 2023. Thank you very much for your presence and contribution of extraordinary thought over these past 2 days. And I apologize if there are any mistake that I said or did, either intentionally or unintentionally. Once again, I'm Dede Setiadi. Thank you very much and see you on the third International Seminar on Fish and Fishery Sciences 2023. Good afternoon. Wassalamualaikum warahmatullahi wabarakatuh. Om Santi, Santi, Santi Om.